So welcome to this class. This is a recording from our Wednesday afternoon live online session. So I typically try to organize these opportunities for us to have a little bit more interaction that's more synchronous in nature than what we typically have in these asynchronous online classes. And one of the ways of doing that is through these optional live online sessions. So for that reason, I will be also recording this session, and you will be able to watch a recording of this session at the later time at your convenience if you feel that that's something that you benefit from. And at this time, I should be sharing my screen, which should allow you to see the kind of a little presentation that I've prepared for our afternoon session. We did meet earlier on today for a morning session, and during the morning session, I took time to go through the really basics of when it comes to the material that we covered on week one, kind of a crash course of that material. I do want to emphasize, though, that your textbook is the main source of learning material, and I provide a variety of other materials to supplement that as part of the Canvas course, but this is simply kind of like an condensed version of the key points of what we should cover and what are the big take-home messages from this material, but by no means not an exhaustive list of all that. So on this session, I wanted to shift our focus to the material that we're focusing during the week 2 of the course. So this week, I wanted to make time for us to review something that I think is largely just a refresher for many of you. You've covered these themes, these topics as part of your introductory biology classes. So this is just a chance for me to make sure that we all find ourselves at the same place as we move onwards with studying various body systems, so that we have a shared understanding, shared baseline of what you should know about tissues and what are the key concepts with that. I will also make a little bit of time today, provided that time allows so, for having a look of the homework, so what to expect in terms of that. And I also want to take a moment to kind of look back or look forward, actually, and see what's coming up next week. So let's get started. Typically, my sessions when we meet include some kind of a self-care opening touch point activity where I really want to find out how is everyone doing and how are we doing as a group. I do realize that it's a little bit harder if we don't have a great range of attendance. Like today, I'm here by myself. So rather than checking in and asking how things are going in this format, I want to take this moment to encourage you that you can always reach out to me. Remember, I offer office hours, I offer these live online sessions, and I'm very, very reachable also through the Canvas messages. That really is probably one of the best ways to get hold of me. I'll be also always happy to make time to meet with you, whether it's in a synchronous setting or whether it's us messaging back and forth to answer any questions, any concerns, any comments. I really want you to know that you're not on your own, even though this is an asynchronous online course. I'm definitely here alongside you and wanting to make sure that you succeed in this class. So, when we're talking about the tissues, we really find ourselves back discussing about the basic concepts that we've covered in your introductory biology course. During the introductory biology course, whether it was Bio 181, Bio 156, or whatever was the course that you used, we reviewed life on various different scales. And I think that we started at the level of atoms, or even having a look of the subatomic particles. We saw how atoms can come together forming molecules. And we also had macromolecules, which were larger, naturally occurring. We discussed some of the most important ones of those. We also saw the level of the cells, the smallest functional and structural unit of life that's capable of existing on its own, so supporting itself. And we saw how cells were made of cell organelles. And today's focus really is to move away from those cells, and we'll get to revisit some of the basic themes when it comes to the chemistry of life, when it comes to the cells that are essential for all form of life, and that more complex forms of life are made, multicellular organisms, and also some of the basics of genetics. We'll get to visit that next week. But today our focus moves one step larger than cells. So we'll be having a look of tissues and discussing about the role of tissues and most important tissues, and kind of establishing a baseline for that, where we should be at the moment. You will remember that when we have multiple tissues working together towards the same goal at the same area, typically these ended up forming organs. And when we had multiple organs that were connected, serving for the same purpose, we developed organ systems. And obviously, living organisms were made of multiple organ systems. So that's kind of to give us an idea of the levels that we're working with during today's and this week's material. Another thing that I wanted to highlight was that cells are very, very small. So typically with a naked eye, it's hard to see cells. You might at the best see the large shape of the cell. I think if you were really lucky, you might see to that level, but we typically need a magnification tool to see more details of the cells. And light microscopes are largely found in most classrooms, most labs, and that allows us to have a basic idea of the what goes on in the cell. So by using various different stains and staining mechanisms, we're starting to see for example, outer boundaries of the cell. We're able to stain the nucleus of the cell. But really with a light microscope, we don't get much further than that. And that's where we recruit even more advanced magnification methods, such as electron microscopes. Rather than having the light to pass through the sample as we have in the light microscope, in an electron microscope, we're actually using electrons and reflection of those to formulate an idea of and magnification of what we're seeing. Typically, these are the results of these images. Imaging doesn't contain color, so we might add coloring later on to these electron microscope images. Typically, they're kind of surface appearance pictures of structures, but it helps a lot when we add that coloring a lot of the time on these investigations. So we really see that both light microscopy and electron microscopy are very worthwhile approaches. Electron microscopes are rather expensive tools, so they're not always available at all settings. And there's a little bit of more skill and technique needed with operating one and so on. So typically on the classes, we stick with the level of light microscopy. Kind of jumping off from this, but to a related topic, since this week, we are talking about tissues. We're talking a little bit of touching about the microscopy and so on. And it's really important for me that we have those authentic laboratory experiences as well. And even though this is a virtual format of the class, I want you to have those. I am trying out something new this week. We're doing a simulation of studying cells or tissues under microscopy, and that whole process of preparing a slide for that. So you can keep an eye of that. I'll try to touch upon that towards the end of this session. But all of these things that we cover in a week are related to each other through a bigger plan. So when we're talking about individual cells and study of individual cells, we're talking about a field of cytology. But when we're studying tissues, which were essentially made of multiple cells on the same area, shared functions, shared characteristics, we now move to the field of histology. So I just wanted to make sure that we're clear of that definition and that difference between the two as we're starting this journey together. So let's talk about tissues a little bit, what we need to know about the tissues and what's kind of the general gist that we want to start with. So tissues are made of groups of similar cells. The structure of the cells is similar, and these cells typically share also a common or related function in terms of what they do, what they exist for. And we often end up finding that tissues that we're studying are originating from the same area. Obviously, you can have tissues in many different body areas, but when we're studying of a sample, typically it's collected from one area as well. So the study of tissues, like I mentioned, is going to be a field of histology. So when we talk about tissues and when we're kind of considering this theme, we can really divide four large, generally well accepted main groups of tissues. There is a lot of variety amongst each of these groups. So this is just a broad strokes, big picture view of the tissues that we'll be covering. But I think that it provides us a good starting point for what we're discussing. So these four main groups that we'll see are going to be epithelial tissues, connective tissues, muscle tissues, and nervous tissues. And I really want to provide you a really quick overview of each of these first, and then we'll talk about each in a little bit more detail, even though still we have to keep it kind of a surface level for the interest of time. Your textbook will provide even greater level of detail. So when we're talking about epithelial tissues, some of the examples that come to mind, and I think that really help us to illustrate what epithelial tissues are, for example, our skin. So we find epithelial tissues lining our body's outermost layer. We find epithelial tissues also in many other body parts. One that I've highlighted here is the lining of the digestive tract. So digestive tract is basically just one continuous loop starting from the mouth, ending to the other end, and just a tube that continues through your body. At certain parts it becomes wider, certain parts more narrower, certain parts we're adding certain chemicals, liquids, we're processing the material inside, but all of that is lined with a tissue. So when we're talking about epithelial tissues generally form various different boundaries between parts. So for example, the insides of your body and outside world are kept separate with the skin. So we're seeing this concept of boundaries in that sense. So really these are protective layers for structures beneath them, keeping, for example, pathogens outside and so on. Another thing that we see with epithelial tissues is that there's a need to have movement of materials across these layers, but it needs to be a selective movement. So how we're able to do that is that we have some amount of absorption. So things can go through epithelial layers, but that's true controlled mechanism. Similarly, epithelial tissues often also are able to secrete materials. So one that really stands out would be, for example, the sweat glands on the surface of the skin. We're constantly secreting materials as well. When we think of that example that I mentioned about the lining of the digestive tract, we're seeing many secretions being put into that material that moves along the digestive tract at different parts of the digestive system. So that's another way of approaching this. And obviously, epithelial tissues can also contribute to the process of filtering. So materials can move through both directions, but we are having that selective ability with that to make sure that we're only or we're able to have some sort of a control over what moves through. Let's have a look at connective tissues. So connective tissues is a really large group of materials, but by large, these tissues do what it says in the thin, if you wish. They connect. And this connection can be either physical connection or it can be physiological connection. So some of the really easy examples that we can think of when we think of the connective tissues are, for example, our bones. They give us our scaffolding, the structure. They help muscles to connect on two ends and typically run over a joint. And that's how we're able to create movements as the muscles contract and relax. Kind of piggybacking on that idea, I think another good example of connective tissues are our tendons that connect these muscles to the bones. So again, we're seeing that very concept of connecting structures. But connective tissues can be far more varied than just what that description would make us think about. So a lot of soft tissues, for example, fat would be an example of connective tissue forming this cushioning around the body. We also see that blood, interestingly, is a special type of connective tissue. And if that feels a little vague at the beginning, think of it this way, that blood helps us to connect different body parts by transporting materials among it. So we're really seeing that connection theme happening again. So really broadly speaking, connective tissues provide us support, they protect us, they bind structures together. The range of connective tissues is really broad, so there's a lot to look at there and a lot to discuss about there. Let's have a look at the next group of basic tissue types, and these are going to be our muscles. So really, if you wanted to give the most simple answer that what muscles do, it is that muscles contract. And by contracting, they, for example, create movement. As a byproduct of this, we can also produce heat as we convert energy from one form to another. Heat is always produced as a byproduct, and that can sometimes be intentional. For example, when we get shivers, so those muscle contractions, in that case, are serving that purpose, not really of moving at large. But if we really wanted to summarize, I think that the contraction is a good place for us to start thinking about when we talk about what muscles do. Muscles are not all the same. So we have many different types of muscles that we will end up seeing. A lot of time when I talk about muscles, students immediately think of skeletal muscles, those big muscles that create movements in the body. So we will get to review those in quite a detail later on this course. So we'll spend some time on that. There are other types of muscles, though. There are, for example, cardiac muscles. So the muscles that muscle that we find within the heart and only at the heart. And we'll reserve the discussion of the cardiac muscle for your BIO202 class, where we get to look at that in a greater detail. There's also a third group, smooth muscle, and smooth muscle is typically found in the intestines, regulating the passage of materials, but also around blood vessels, regulating the diameter of the blood vessel. So we're going to see again more detail about that in your Bio 202 course when we talk about the cardiovascular system, and we'll also get to talk about the digestive system. And final group of the tissue types that I wanted to mention are going to be our nervous tissue. Nervous tissue at large, you think of things like our brain, our spinal cord, and various nerves that we have running in the body. All of these work towards the same goal of facilitating internal communication. This communication can be towards the central nervous system, typically sensory functions, carrying information to the central nervous system, or it can be away from the central nervous system, typically activating some kind of body responses, body movements, and so on. I'll reserve the nervous system towards end of our time together during this semester, so I'll keep it really short today, but you'll get to hear a lot more about it. We've got quite a bit of time reserved for talking about different parts of the nervous system. So what I want to do next, I want to review each of these 4 types of tissues in a little bit more detail for each one. And the first one that we are going to start with is the epithelial tissue. Often you will see that rather than talking about epithelial tissue, it might be that we also refer to the epithelium. So there's really 2 main types of epithelial tissue that we see. We have types that are more focused on the forming the coverings and the linings along the body. So we kind of had a look of those already earlier on. Things like skin, it would be a good example. So really forming these boundaries between external and internal surfaces and so on. But we should not forget that epithelial tissues also include glandular epithelia. The example that I've highlighted here is the salivary glands. So we have various different glands that secrete materials. And these secretory tissues that we find in the glands are another type that falls underneath this category. So I wanted to highlight that. When we think of the functions of epithelial tissues, we end up seeing that there's a broad range of what they can do. They provide us protection, they help with absorbing materials, also filtering materials, exerting materials, secretion, sensory reception. So there's a lot going on with epithelial tissues. And really, when I think about it, this is going to be our first proper chapter that we'll have a look in a little bit of a time when we start talking about integumentary system. So the skin and associated structures such as hair, nails, glands, and so on. So that's when we are going to spend a lot of time talking about epithelial tissues, but also other tissues that are associated. Onwards, we can classify epithelial tissues in 2 different ways. We can classify epithelial tissues based on the shape of the cells that we see. And what I've drafted in this little table that I put together, we can see kind of a cross-sectional view of cells. So the first group that I wanted to talk about were squamous cells. Squamous cells are these kind of flattened disc-shaped cells if you look them from the side, and they typically have a central nucleus, but they look a little bit, if you want to be imaginative, like fish scales. So they're really just flattened cells. These are ideal for allowing the passage of materials back and forth through these because the distances are not great. They do provide some level of protection for materials underneath and often serve for forming a surface along which material can also pass against it. If I think of digestive system, we see, or cardiovascular system, the vasculature, we see that when it's a nice smooth surface, the movement of material is easier in comparison to when we go to, for example, capillaries that have non-continuous surface in the vasculature, and that then slows the speed of flow of the blood past these squamous cells. It facilitates a greater range of exchange as well. So I often talk to my students about, think of when you're driving on I-10, when you have a nice solid asphalt or when you have a lot of potholes. The next epithelial tissue type that I wanted to talk about were cuboidal cells. So these cuboidal cells, really, if you look at them as a cross-section, they look a little bit like cubes in terms of their shape. So that might be an easier way to think about them. So they're like a bunch of cubes with a central nucleus located in the middle. So in this picture, we're seeing multiple of them next to each other. And the final type of epithelia that I wanted to talk about is going to be our columnar epithelia. Columnar epithelia look like pillars or columns, if you wish. So they're longer than they're wider. They as well will have a nuclei. Often there might be other adaptations at the end, like in this picture, we see microvilli at the very top, but they appear these pillar-shaped cells, often packed tightly, neatly next to each other. So whenever we just have one single layer of epithelial cells, may it be squamous, cuboidal, or columnar, we talk about that tissue being simple. So simple refers to one layer of these epithelial cells. When we start to get multiple layers of these cells packed on top of each other, we move on to talk about stratified epithelia. So we can again use the same language that we just learned, but we use now the term stratified. So in this first box, we're seeing stratified squamous epithelia. So we're seeing that there's multiple of these flattened disc-shaped cells packed on top of each other, forming a thicker layer. So it's multi-layered on top of the basement membrane. This is something that I think a good analog of a stratified squamous epithelia that we end up seeing would be, for example, when we discuss about the skin. The outer layer of the skin is actually dead cells, and new cells are constantly being made next to the basement membrane. So we have these multiple layers that give skin quite a bit of durability, quite a bit of protective functions, and it's constantly regenerating in that sense. If we have a look of a stratified cuboidal tissue, we see the same concept that we saw before, that we have these cube-shaped cells, but in this case, we're seeing multiple layers of them on top of each other. So we're starting to see that it's not just one cell, but it's multiple cells on top of each other. So when we have multiple layers of cuboidal cells, we start talking about stratified cuboidal epithelial cells. So I hope that that helps to kind of draw a difference or understand what's the big deal of separating these shapes of the cells also based on the number of layers. And the final type that we can work out with the same logic is going to be our stratified columnar epithelial cells. So here we see these more of a pillar-shaped cells, but we see multiple layers of them on top of each other as well. So again, forming a thicker layer, more durable layer, if you wish. So I think that this table is a good place for us to start, but I want us to go a little bit further. So we will actually end up seeing that these are not always as easy to classify the tissues that we'll see on microscopy. We sometimes have tissues where it appears almost as if there would be multiple layers of cells. As you can see here with the nuclei of these columnar cells being kind of on a varied levels of plane. So it looks like it would be multiple layers, but when we pay attention, each of these cells actually connects directly with the basement membrane. So in reality, it's just a single layer, but it looks like multiple layers. So we call these kinds of tissues pseudostratified tissues. So you can think of that through the word pseudo. Pseudosciences are fake sciences. So sciences that we don't have hard evidence for. People believe in things like reading tea leaves. So it's not really a real science. It's a fake science. So similarly, these pseudostratified tissues fake like they would be multiple layers, but in reality, they're just a single layer since each of these cells all connect directly to the basement membrane. Let's continue our review and let's talk a little bit about glandular epithelia. So glands are cells that produce and secrete fluids, and these fluids can vary quite a bit based on the purpose. And we can classify these glandular epithelia in two different ways. First way that I wanted to classify them was based on the site of release of the materials. So we have something known as endocrine glands. These are glands that secrete hormones, and they release these hormones to the cardiovascular system, to the vasculature, so blood vessels, and there these hormones travel to all parts of the body. So we'll get to talk about more about endocrine system as your very first chapter in BIO202. That's what I always start with my BIO202 classes. It is kind of a challenging chapter, but it's a fascinating chapter. The second type of gland type that we see in terms of the site of release are exocrine glands. Exocrine glands are glands that are located at the site where they act. So whereas hormones from the endocrine glands can be released anywhere, you don't have to have the gland next to the structure where it acts because those hormones travel in the bloodstream to all parts of the body. But exocrine glands are very, very local. A good example would be salivary glands. We have three main types that we'll end up seeing on BIO202, and they are located around the major extrinsic salivary glands, around the mouth, because that's where their secretions are heading. So they're localized close to the site where their secretion is needed. They're also typically characterized by having a duct. So exocrine salivary glands have ducts that open then very close to the glands, to wherever the secretions are needed. In case of the salivary glands, they open to the mouth. In case of sweat glands, which would be a good example of exocrine glands, they have a duct that opens right on top of the gland on the surface of the skin. So one way to divide these glandular epithelia into two groups. That's not by any means the only way of classifying glands. We can also talk about the number of cells that we have when it comes to these. And let me just jump here to correct my slide. So we can have these glandular epithelia that is made of just one cell or they might be multicellular, made of multiple cells. So another way to draw this classification. And again, your textbook goes into quite a bit of detail, providing you different examples of these. So let's move on to our second group of major tissue types. And these are going to be our connective tissues. And like I said, the group of connective tissues is incredibly diverse. So when we are thinking about the types of connective tissues that we really have a great number of options there, what they do, what their functions are. So connective tissues, like I said, connected body parts, either directly, physically, or physiologically. So we see that these are responsible for things like binding structures, supporting, protecting, insulating. I think that that's a good example. For example, adipose tissue, fat tissue, provides us insulation. Storing reserve fuel, again, adipose tissue, a good example of that, or transporting substances, as we saw in case of the blood. So it is not just the bones and tendons and ligaments, even though those are important connective tissues. The group of connective tissues is extremely varied. And there's really four main classes of connective tissues: connective tissue proper, cartilages, bones, and then blood. So you can learn more about each of these four main subgroups, again, from your textbook, but I at least wanted to highlight them for you. What is common for all connective tissues is that they all share this ground substance, presence of fibers, and cells. So those are just general characteristics that we see with the connective tissues. Let's have a look of our third group of tissues, and this is going to be our muscle tissues. Like I said, sometimes when you ask from people, one way to look at the function of the muscles is that their main function is generation of movement. We saw that there's actually three different types of muscle tissues. So those skeletal muscles are the big muscles that move body that we often end up thinking of. And I think I have a very good example here of a collection of some of the most important skeletal muscles. And you'll get to revise these later on this course. We'll spend time learning the most important ones. So that's something that we will end up focusing on. But at this point, I just want you to kind of make a mental note that that's only one of the three types of three types of muscle types that we have, these skeletal muscles. Let's keep going further, and let's talk a little bit about cardiac muscle. Cardiac muscle instead was found only within the heart. And cardiac muscle, unlike the skeletal muscle where we get to choose that do we create a movement, is not under voluntary control. So it will automatically contract as we have the heartbeat. So we don't really need to think about, now I'm going to have a heartbeat. It will automatically happen. And finally, smooth muscle. I mentioned that earlier on real quickly that we find smooth muscle in the digestive tract, in the intestines, controlling the passage of the materials through this continuous tube. And we also find smooth muscle around the blood vessels, controlling again the diameter of blood vessels, whether we need to more flow to a side or restrict flow to a side, or through this control of the diameter have an impact also on the blood pressure within the vasculature. What I wanted to do next, I wanted to show a little bit of more of a histology on a microscopic scale, a single cell or few of these cells of three different types of muscles that we have in the body. So first of all, the skeletal muscles. Skeletal muscles, you remember, they were responsible for these large muscles that generate movements in the body, and they are made of on a cellular level of single cell. And these single cells are kind of cylinder-shaped cells. They will have multiple nuclei, and typically these nuclei are located at the edges or at the not in the middle, more on the edges of these skeletal muscle cells. There's going to be multiple of those, and for some of you that might sound confusing, because didn't we just learn when we talked about the cells that every cell had only one nucleus? That is true, but during the process of development of the skeletal muscles, actually multiple cells fuse together to form this structure that you'll end up seeing then, which has multiple nuclei. So they're multinucleated cells. Another characteristic that we see with the skeletal muscle is these striations that run through these cells. And we'll get to see about these striations in a far greater detail when we talk about the muscular system and structure of a muscle and a sliding filament model and all that microscopic stuff a little later on this course. I did want to highlight that skeletal muscles, so these muscles that generate big movements around your body, they are under our voluntary control. So we have a great amount of control over that, which movement we do, how do those muscles contract, and through that regulating our body. So they are under voluntary control most of the time. Let's have a look at cardiac muscle. So cardiac muscle, we realized you find it only within the heart. And if we look at these cardiac muscle cells on a cell level, these cells are actually branched. So they connect to multiple other cells of other cardiac muscle cells. And this kind of makes sense. It makes sense because we want to have all of the cardiac tissue on the same side contracting all at once. If these individual cells that form the cardiac muscle tissue all did their own thing, we would have really hard time creating a synchronous cardiac contraction. So they have this branched appearance. And at the end of these branches, we have something that's known as intercalated discs. So that's another characteristic that might be worthwhile to add there. I'm gonna add it onto these notes. So that might be a good thing to also make a note of. We also see that these cardiac muscle cells, they do tend to have only one nucleus, typically located very centrally, and a lot of striations. So another microscopic characteristic that we can see. I mentioned these intercalated discs. I just want to expand on those just really briefly. These are adaptations at the end of these, where these cardiac muscle cells that were branching connect with other cells that allow easy passage of materials and messages from one cell to another, again feeding into that concept of making sure that these cells on a same area contract nicely together. Heart does beat automatically. You do not need to wake up and think in the middle of the night, I should have a heartbeat now. So these cardiac muscle cells are involuntary. So that's worth of highlighting of them as well. Let's keep going onwards and have a look of smooth muscle a little bit. So smooth muscle was found within the intestines, around the blood vessels, regulating vessel diameter. On a cell level, if we look at a single smooth muscle cell, these are kind of spindle or blob shaped, depending whether they're contracting or relaxed. They have one nucleus that's very central, and we don't find any striation within the smooth muscle. And again, smooth muscles are involuntary, so we don't need to think about I'm going to dilate or I'm going to constrict my blood vessel diameter, or the same thing for the digestive tract. It just automatically happens. Moving onwards to the last tissue type that I wanted to mention, which is going to be our nervous tissues. Nervous tissues are responsible for regulating and controlling body functions, and some of the most important components of the nervous system contain, for example, your brain, your spinal cord, and various nerves that then we see around the body. And we'll get to talk about these in a great detail towards the second half of our course. I do want to mention at this point that not all nervous tissue is made of same cells, though. We have this general idea what a typical neuron looks like. And in reality, neurons vary greatly from one neuron to another based on their functions, locations, and so on. But neurons are not the only part of nervous tissue. In addition to that, we have something known as neuroglia or glial cells. These are more of supporting cells that support nervous tissues, neurons. And we're finding out these days, for example, that a lot of research has been dedicated on actually studying the nerve cells, neurons, but neuroglia is actually incredibly important. Some of the leading Alzheimer's disease that we're doing is indicating that actually the involvement of neuroglia or deterioration of neuroglia might be really important on these conditions as well. And that really brings us to the end of that kind of a review of the key concepts to highlight from the chapter 5. What I wanted to do next was to spend just a little bit of time having a look of what you can expect from the homework for this week. So let me jump out from this presentation. And what I'm going to do instead is share my screen, but let me get to the right screen that I'm sharing. So at this time, you should be seeing my Canvas course for this class. And we are now on week 2. So we're looking at the chapter of tissues. And let's have a look of what all material I have available for you. I have those learning outcomes. I have some terminology that you should walk away. And I have a couple of lecture videos providing really brief kind of introduction to some of the key points that you should take away from this chapter. So that's there available for you to supplement your study of the course textbook, which you have in an electronic format. I've also uploaded PowerPoint slides of this chapter because some students really enjoy using them, making their own notes there or finding them as a good supplementary material when they're studying the material. What I'm asking you to do this week is found underneath the heading of homework. And for the homework, I have 3 pieces of assignments that I'm asking you to do. And I'm asking you to make sure that you complete them by the end of this week. So end of this week is on Sunday, which is, I believe, September 3rd. 1 minute before the midnight is when all of these assignments are due. So I wouldn't leave it to the very last minute. I would work through them, giving yourself time. Remember, with my course, you can take quizzes multiple times. You can, the best grade that you get is what counts towards your final grade on those when I do those calculations and so on. So what I have for you is a quiz. This quiz really contains questions such as multiple choice, true, false, matching, drag and drop assignments, things like that, that will help you to kind of grasp some of the key concepts of the chapter material. In addition, I'm asking you to complete a practice anatomy assignment. Again, this carries points that add up towards your final grade. And practice anatomies are more of usually visual based, picture based questions of the material. So especially when we get to actual organs and so on. So you'll be labeling structures, naming structures. On this week's practice anatomy, since it's more of kind of a tissue type of a chapter, you'll be, for example, dragging and dropping elements, statements, terms to right categories, naming parts of things like that. And the final piece of homework, which you didn't see last week, which is something that we see for the first time during this week, is a simulation. So simulations are something that allow us to have a little bit more involved experience of what it is to be in a lab, even though we're doing this course virtually. I'm trying out these simulations for the first time during this semester, so I've made sure that they don't carry a huge number of points, even though there's a fair amount of work to do those simulations. But I just want to start easy and get your feedback, get your experiences, and take it from there. So by no means don't feel that they're worthless activities, even though they don't carry a huge amount of points. This is the first time I'm enrolling or utilizing them. So I want to make sure that before I go and make them high stakes activities, I give you a chance to provide me feedback. And this week's simulation is all about how we would prepare a microscopic slide. And in this case, we're preparing this microscopic slide from a bacterial sample. So this simulation will allow you to have the very same experience that one would have if they were taking the class with me in person in a lab setting. So those are the 3 assignments that I'm looking for you to submit. And we can have a really quick look of each of those in just a second. But I do want to point out this final section that I have. And I think I referred to this already in our week 1 material, but I just want to make sure that you are clear what I'm doing here. So sometimes I get students who want to go beyond the minimum. They want to learn a little bit more about the topic. They want to tackle more of application-based questions, or they want to tackle more clinical-based questions of the material. And I do want to serve those students. I do want to offer them an option to do so. So I've developed these want more sections. These are completely optional. They're not required as part of your homework assignment. The want more section provides you kind of a rather nice summary of the chapter content, some of the key points listed. So a different way of revising. Have you gotten what you need to get out of this? I have some discussion-based questions. If we were probably in a classroom, we would have more of discussions about these. But you can use these as prompts for you to check your own thinking. And if you want to put it down in writing, you can absolutely do so. And hold on to that thought. What do you do with that writing that you might do if you feel like it's helpful for you? I sometimes have students who are really interested about a particular topic. So I have really good books, articles, available for you regarding the topic. So in this case about tissues. So if you want to go and do additional reading, some of them are in an electronic format. Some of them you can just Google. Some of them are physical textbooks that you can, for example, find from the library. I also have critical thinking questions. These critical thinking questions really allow you to take what we've been talking about and apply and challenge yourself when it comes to these questions. And again, you could note down your thoughts as you're working through them. You don't have to do all of them. You could just do one. You could do whatever you are interested, or you could do all of them. These are completely optional, not required. You can get 100% in the assignments without ever doing these. And finally, I try to always have a clinical application question of the chapter material. So I have one in relation to this tissues chapter as well. So some students really enjoy tackling these and having a kind of a different think about it, having a think about it. How does this material lend itself to when we're studying the clinical aspects of the course material? So you find all of this as this want more section on our learning module. And then I have a submission space. If you did write down any answers or any notes or did bullet point notes or however you wish to tackle these questions, you can submit that work for me here. It is not required. It's not something that you have to do. But if you want to gain extra credit points, that is one way how you could do that. And you can either upload a file or you can write your answer as a text, whatever you prefer, and then submit it for me. So that's something that's available for you as well. Like I said, not required, just there in case you want to go above and beyond. But these 3 assignments are what I'm looking for from you for this week. I do sometimes get students asking, what will happen if I don't do them? Well, it will really basically just end up affecting your grade point accumulation for this course. If you do miss a deadline, do contact me and we can have a discussion if I could extend that deadline for you. It's not automatic, it's case by case basis, but that's something that I'm sometimes willing to do. So again, contact me if you miss any of these deadlines. But since this is an online course, I really encourage you to try to stick with those deadlines. If you do want to work ever ahead of the time, you can totally do so that. I have all of the learning modules already open for you and available for you. For example, if you know that you're ever busy, that material is there to help you to make a kind of a head start for those weeks when you might have less time to focus on this class. But these were the 3 assignments that I'm looking for getting back from you. And I'm going to stop sharing my screen just for a second. And the reason why I'm stopping sharing it is that I do want to show you what happens in these different assignments, but sharing that, I have to go to the publisher's platform in a little different way than how you go. So just for the sake of privacy, I'm not going to share that process with you yet. It's a little bit of a different deal than what you guys do. And let me share my screen again. Now I'm on our chapter 5 quiz. So we can have a look of, we're not going to go through all of the questions, but have a look of what you can expect that might come up in terms of that. So you should be seeing my screen. I know it's not ideal for some reason. When they did updates, the screen is kind of squashed to the side, but it's enough for us to have a look of what to expect in terms of the quiz this week. This week's chapter 5 quiz is made of 12 questions worth of 10 points. And for example, here we see the first question, which asks you to list the 4 primary types of tissues. And then from the options that you are offered, you should choose the correct one. Like we just saw, the 4 main tissue types that we see were epithelial, muscular, connective, and nervous tissue. You will also have a reference to where in the textbook you can find this answer if you're wanting to do that. You have a few other questions here. We're being asked to choose from multiple choice again. Which of the following terms describes a thin slice of tissue mounted on a microscope slide? This would be a histological section. So again, you get to learn more about that. You will have an option to click on hints, and that hint will provide you kind of guidance if you get stuck with any question and you don't quite know what to answer. So it will talk you towards the right question. And again, I remind you that you have your textbook. Textbook is the primary source of material, so you can have the textbook open at the same time as you're taking the quiz if you prefer to learn that way. I'll show a couple of more picture questions and then I'll leave it for you. Here, this question asks you to click and drag each label into appropriate category according to which type of epithelia are we talking about. And let me see if I can move this picture on the side. So here we have options simple and stratified epithelia. We saw that in simple epithelia, every one of those cells touched the basement membrane, so that base layer. So there was only one layer of these cells. Simple epithelia is ideal for allowing really quick diffusion of materials. So across this single layer, it doesn't take as long. And we have also simple epithelia lining things like the innermost layer of various different tube-like structures, such as renal tubules of the kidney. Then we have a question about stratified epithelia, and we see that on stratified epithelia we had multiple layers of cells. So some of the cells will rest on top of the other cells, so multiple layers. These are really ideal type of epithelia to provide us protection for any damage on the surface because we have multiple layers, so even if one layer gets damaged, we still are protecting the deeper layers. We find stratified epithelia, for example, on the superficial surface of the skin. So we talked about that, how skin was made of multiple layers, actually the outermost layers being dead skin cells. So again, we're seeing this concept there. We find stratified epithelia also at the sweat gland ducts. So they're made of these ducts that have multiple layers of cells in those, and sometimes stratified epithelia can be keratinized. So keratin has built up, as we see, for example, keratin in the nails, hair, the outer layers of skin cells sometimes, and so on and so on. So again, this is kind of a different way for you to tackle the material. You drag each of these statements to the right box and approach the questions based on that. Let's have a look at one more example. This example is a multiple choice example. You're being asked which of the following statements is false about epithelial tissues. So all other statements are correct except one. Well, we see that epithelial tissues function in absorption. That was one purpose. That was correct. Epithelial cells can be tube or columnar shaped. We saw that. That was true as well. Epithelial cells do not secrete a ground substance. That would be true. And epithelial cells are typically arranged in layers. Sounds correct to me as well. So the only statement here that wouldn't be true would be the statement that says that epithelial tissue is composed of cells and a thick vascular matrix. That does not sound correct and apply to what we're talking about epithelial tissues. Again, you would have a chance to get hints if you're kind of stuck that I don't really know how to approach this question, and you can even find at what part of the textbook it is covered, this question. So that's an example for you of what to expect when it comes to the quizzes for this chapter. And again, I'm jumping away for a second because I do want to show you, give you a taster of what to expect when it comes to the practice anatomy assignment for this chapter. And again, let me share my screen. And here we see that chapter 5, practice anatomy, contains 10 questions, worth of 10 points in total. Let's have a look at a couple of those questions. So the first question is actually you clicking and dragging labels to correctly identify different sections that we're seeing on the pictures. So here we're seeing a longitudinal section of a bone in this case. So you would drag that there to highlight that. And we see that longitudinal section here cut the structure in a lengthwise through the specimen. Then we're going to see a cross section next. Here, this cross section shows you that this plane is also known as a transverse section. It cuts it, showing kind of a horizontal view, if you wish. And finally, we had the oblique section. This is if you looked at the morning's video that I've recorded for you. Sometimes, even though we have these three planes that we had in the body, X, Y, Z, if you compare it to the mathematical way of creating these planes in geometrical illustration of structures. If it doesn't align with any of those three planes that we generally use, we talk about oblique sections. So oblique sections are kind of in a weird angle in comparison to what we normally do. We might use these, for example, when we talk about the study of the heart. Heart is located in that weird angle in the chest, so we might need to take that blade through it. Let's have a look of another example. So here you're identifying each of the tissue types, dragging the label that describes it, and let's see what we can see here. So here we're seeing a lining of the ducts of the sweat gland, and I'm trying to pull up the further explanation. So we see, for example, here aiding in temperature regulation. So I think that this is a diagram of sweat glands duct. You can see the cells there and the center of the duct there. Here we're seeing another, this is a microscopic image, not a drawing, of covering the surface of the skin, again helping us to protect ourselves against pathogens. So that's a good example that we are seeing there. Here we're seeing a further lining the internal surface of the urinary bladder. Here on this diagrammatic representation, we're seeing the lining of the small intestines, which really helps with maximizing the absorption. So again, these pictures are gonna be from your textbook. Use your textbook to guide you. It gives you a chance to kind of practice in a little bit of a different manner. Let's have a look at one more example. On this example, you're being asked to determine which connective tissue type in each image below represents, and then click and drag the boxes to label them. So let's have a look of those. So here we're seeing reticular connective tissue, which provides support framework for the spleen and is found, for example, in the bone marrow and lymph nodes. So again, looking at this histological actual microscopic image from your textbook, you will find guidance and it will help you to understand. Here we're seeing on the background areolar tissue forming basement membrane of the epithelia, found in fascia that holds the muscles together and forming the serous membrane lining of the lungs. And the final picture that we're seeing here is a histological view of the adipose tissue, so fat tissue. This is where energy is, for example, stored and provides thermal insulation as well. So these are the kind of questions that you can expect in the practice anatomy. And the very last thing that I wanted to share with you before we wrap up for today is what to expect when it comes to the simulation. And let me see if I can demonstrate it. It might be that I don't have access once I've opened it to demonstrate. Unfortunately, I don't think that I can pull it up at this point anymore. But basically, what you will see is simply a chance for you to kind of go into a virtual lab setting, and you'll be preparing your own microscopic sample. In this case, the sample is actually from a bacterial source. We've grown bacteria. There's a few different ways of growing bacteria, either on a agar blade or on a liquid liquid broth, and then you will prepare a microscopic slide of that, stain it to make structures stand out. And as you're doing that process, you'll learn a great deal of skills, great deal of information. So many students generally like doing these activities. They like doing them, especially in a lab setting. But since we cannot be in a lab setting, since this is an online class, I want to offer it at least now in this virtual setting for you. Remember, like I said, I am trying this out and I'm looking forward to receiving feedback. So later on the semester, I'll be collecting your feedback, thoughts, whether you are liking these simulations or not. So I think that this really brings us to the end of what I had intended to share. Let me just jump to one more view and then we're calling it a day. So at this time, you should be seeing my PowerPoint again. And we have done really everything I set out to do this week. Only thing that's left is the reminder of make sure you do those 3 pieces of homework. One more is not required, but I do ask you to do the quiz, practice anatomy, and that simulation. Please do them by the end of the week. So 1 minute before end of Sunday, I'm looking for them. And what we'll do next week will be a bit of a kind of a review of things that you already know, even in a greater scale. We'll cover like a crash course of introductory biology course when it comes to what you need to know to succeed in BIO201. So we'll have a chemistry of life, look of chemistry of life, we'll have a look of cells, and we'll talk a little bit about genetics as well. So this covers everything that I had intended to cover today. If you are unable to join me on these live online sessions, it doesn't mean that you wouldn't still receive help. You can always join me during the office hours. I'm available for you on Monday and Wednesday mornings, and you can always drop me a message through Canvas. If you want to have synchronous discussion, we can do that, or you can just message me and we can exchange messages. But I am here to help you to succeed. I know that sometimes taking an online class might feel like a lonely place, but that is not what I want you to have an experience of. I want you to know that you're supported every step of the way, and I am here to make sure that you can reach your goals for this class. So I'm going to wrap it up at here. Like I said, don't hesitate to reach out to me, and this concludes our live online sessions for this week. I hope you have a very good week, and let me know if there's anything I can help you with. Bye.